Thank you. Good afternoon, everybody. My name is Aamir Mecklai. I'm Managing Director at BofA Healthcare Investment Banking, focused in biotech. I'm joined here with my co-panelist, co-moderator, Dr. David Ting from MGB. We're very fortunate to have this set of esteemed group of panelists join us today. They're all at the front and center of the next wave of oncology research. Clearly, there's been an avalanche of oncology drug development in the antibody space for immune-targeted therapies and combinations. Recently, we've seen rapid advancements in development, both clinically and regulatory in the space of bispecific antibodies. By my count, there's probably been over a dozen or so that's been approved by the FDA so far, with hundreds behind them in the pipeline for clinical trial development. So let's kick off to just set the stage here. Where are we seeing these therapies work, and how are they building upon the successes of established immunotherapies, and what have been some of the challenges in the development of those drugs? So I guess I'll ask Roger to kick us off there.
Yeah, thank you. So I think there's been a considerable amount of interest in the development of targeted therapeutics using bispecific approaches. And fundamentally, there are two aspects to that. The first is finding an appropriate ligand that is reasonable to target, and then the second is what's the warhead. Oh, there's a linker in between, by the way. But those are the sensible questions. And there's, while not an infinite range of possibilities, it's quite large. There's an enormously large set of potential targets, particularly because there are certain cell populations that one can reliably deplete without causing serious injury. It hasn't been possible to get truly tumor-specific ligands to address, but there are some that are tumor predominant, and that's been helpful. And by juxtaposing either a cytotoxic drug or an engager that can link to the T-cell antigen receptor, particularly for cytotoxic T lymphocytes. Of course, they all share the same T-cell receptor, but one wants to engage the cytotoxic T lymphocytes in most cases. Or a radionuclide, one can have a fairly dramatic effect. And the therapeutic window for those things, well, it's a little hard to estimate at the beginning, but there's been enough improvement in therapeutic window, at least in some of those circumstances, that you actually see a beneficial response, and that's led to the registration that we see. How large is that universe? It's potentially very large. And we still, I think it's fair to say, don't really understand the rules that govern how you select fundamentally the target A or the ideal warhead B, and any other physical properties of the antibody drug, antibody linker, or antibody nuclide complex. I think all of those things are potentially in play. I guess my only contribution to this field, if there's any, is having brought into the practice the first of them, which was Blincyto, which was at that time what we called as a bispecific T-cell engager, or BiTE, and the BiTEs are still very much in the news and being pursued by my former colleagues and previously unknown colleagues at Amgen. And in addition, there are leagues and leagues of other companies that are doing this kind of work, and frankly, it's quite promising. And there are aspects of the modulation of target performance that are unexpected and could turn out to be enormously beneficial. I guess I would turn to a fellow panelist and say, David, you do this for a living. I'm just a commentator on the field and not an especially informed one, but this is what you do.
Yeah, building on what you said, clearly there's a wealth of targets, and I'm gonna shift our focus from the hem-onc where we can eliminate B cells fairly safely and move into solid tumors. Tumors have done such a great job of hiding from the immune system. Everything that they're about is, there's nothing going on here to the immune system, move on by. So as you start thinking about targets, there are going to be a few. We've seen a few already where they're very unique and specific to the tumor and not in healthy tissue, but the vast majority of targets that one wants to go after in these bispecifics are also expressed in healthy tissue. And that same immune response that you're looking to drive that anti-tumor effect with your bispecific, when you trigger a T cell, bridge it to the tumor cell via that bispecific, that same pharmacology biology is occurring in healthy tissues. You're just triggering an immune response. So Janux took an approach where we try to take advantages of differences in the tumor environment. There's an excess of proteolytic activity for a wide number of reasons. And so grafting that with masking, what we've essentially done with our programs, and we have a couple in the clinic right now, we're able to more specifically target the tumor, get that anti-tumor effect, and thereby spare the immune response in healthy tissue. So we view our technology building on the shoulders of the early Blincyto work and everything else that the field has been doing over the last decade and a half. And really our goal is, can we expand the number of targets really by circumventing that healthy tissue immune response that's really critical. We're starting to see activity with our programs, building on what Roger said, Amgen has continued their great work. We've now got tarlatamab in small cell lung cancer that's moving forward, and a number of approaches. So I think the opportunity is clearly real. There are some challenges that we're addressing some, and other groups are addressing others. But if you think about what our immune system does, it eliminates foreign bodies and entities. And tumors have spent a lot of time figuring out how to avoid that. The bispecifics, BiTEs, T-cell engagers as they're called, really is a recognition element that now allows the patient's immune system to recognize that tumor as foreign and trigger that anti-tumor immune response that we're really trying to get. And so with that, I'll just hand off, but that's how we're thinking about the space right now.
Yeah, maybe I can add one piece to those great comments. My own experience at Johnson & Johnson was with the construction of two T-cell engagers, teclistamab and talquetamab, following a license of a product from GenScript that was formed into the CAR-T product called Carvykti. And so we had the opportunity, in comparing teclistamab to Carvykti, to use good old-fashioned pharmacology thinking to understand what the relative affinities to BCMA in that particular case and CD3 should be, and used a different form of targeting than David, the masking approach, where that balance between the CD3 affinity and BCMA's affinity was critical for tissue specificity. You can imagine that if BCMA, the anti-BCMA, or BCMA is at very high density on a T cell, you don't need very high affinity for your CD3 to present those CD3 tails to T cells very avidly, very potently in collection. If the density is very low, then that wouldn't be the case and T cells wouldn't be engaged. So that sort of thinking led to the design of teclistamab, talquetamab, and subsequently a trispecific. And it does offer some degree of specificity and reduced cytokine release syndrome, which is an interesting build on top of, I think, what a lot of groups end up putting these pieces together without thinking through the specific isotherms that are in play.
It's a good point. I mean, one of the issues that we used to take up a long time ago was whether we could design soluble molecules well enough to eliminate the need for CAR-Ts, right? Because an off-the-shelf therapy that's always available obviously would have great advantage over something that involves vein-to-vein time, the manipulation of normal cells, the introduction of new sequences, and then somehow making those T cells expand. I mean, the expansion part is pretty good, but expand and kill the target. Yeah. If one can do that reliably, in a way where you understood all sides of the complex equation that controls the bispecific interacting on the one hand with its tumor target, on the other hand with an activating sequence on the T lymphocyte, one wouldn't need to have a CAR T, frankly. You could just repeatedly introduce those things, which would be greatly beneficial. If you could control the cytokine release syndrome aspect of it, that's even better yet. So there is a frontier here that's worth imagining, particularly as we recognize, again, returning to the hematopoietic system for just a minute, that one can use these kinds of cytotoxic therapies to treat a range of autoimmune diseases, as has been demonstrated, of course, with the example of systemic lupus erythematosus.
So how do you guys think about, some of these things of like fine-tuning the specificity and the targeting? How do you think about like the next generation of technologies that you find exciting on the frontier, leading edge of this area? Maybe David, we could start with you on this.
We're excited about just the wealth of new information about potential targets that you can start thinking about modulating immune cell biology with judicious choices of cis interactions on the same immune cell. So that's an exciting area. In-house, we also have an interest in autoimmune disease. We've redesigned the underlying T-cell engager to be more reflective of the immune response that one sees in responders to Keytruda, pointing back to Rogers. What you find in those types of patients is you get this expansion of the memory population that then leads to a reservoir that continually replaces the effector cells. And the effector cells, don't want to get too deep, those are the ones removing the tumor type. But in responders, you get this memory expansion. What we've noted, Amgen's published, J&J's published, is you don't get that with the contemporary T-cell engagers. You're really relying on the existing effector population. So some of the things that we're excited about are our approaches, and we certainly have one internally that is separate from our masking strategy, that actually is much more like an adaptive immune response. You get expansion of the memory population, maintenance of the effector population, deeper, more durable responses. And similar to Keytruda, at least in non-human primates, we're not in humans yet, you don't have CRS. Mm-hmm. People think about contemporary T-cell engagers, say, well, CRS is just part of the pharmacology, isn't it? And then it is with contemporary T-cell engagers. We all view that as a PD marker of activity. But if you go back to what Keytruda is doing, it's enabling the same T-cells to engender a long-lived deep response, and they do it typically without CRS. Our own natural response does that. So we're excited about what we think will be the next generation of these bispecific T-cell engagers that are more reflective of our adaptive immune response than what we've seen with these contemporaries. And we think that's going to be an exciting area. And there's a whole slew of mixing and matching other targets that we find exciting.
There's one more area just to mention then that extends in a different direction, is to stay with what Roger said is the principal problem of CAR T, and unreliable times of vein to vein and cost, frankly, is significant of CAR T cells. A different direction one can go is to inject a vector like a lentivirus and effectively do what people refer to as in vivo CAR T. Time will tell if this is practical. Companies like Colonia and others are trying that exactly. You can imagine 1,000 ways for that to go wrong, but point by point, there are problems being solved on each of those many ways. And so it's not hopeless that there's a different kind of off-the-shelf approach relative to a T-cell engager. I wouldn't want to belabor all the ways that might not work, but it would be very interesting if one could get that to work.
And we just saw the Capstan AbbVie deal, based on early human volunteer studies. So very promising, exciting.
I think one advantage of those kinds of manipulations, and David, you may want to comment on this too, is when a T-cell engages a potential target, there is an immunological synapse, right? We're not just talking about tweaking CD3 via one of its subunits. There's a whole set of handshake signals that take place, perhaps a dozen or more. And it's interesting to think about how one reconstitutes something that's more like that, that has more of the regulatory features that are involved in the formation of the immunological synapse, which I think is probably relevant to the fact that you get very potent T-cell killing when you treat with a checkpoint inhibitor without suffering the consequence of cytokine release syndrome or overwhelming immune activation. I mean, indeed, one of the most remarkable things about checkpoint inhibitors, as we began those studies now a long time ago with pembrolizumab, now Keytruda, was how benign it really was. I mean, the adverse experience profile for that, and for anyone, and there are people right here in this row who've used this, it reveals the pre-existing immunity that exists against tumor populations without causing broad spectrum immune activation, and hence it's surprisingly tolerable. And of course, that's where we'd like to get to with some of these soluble manipulations.
This is a question from the audience. In addition to the sort of design of these things and affinity considerations and combination modalities, what are your thoughts about overcoming tumor heterogeneity and also the suppressive tumor microenvironment? How do we overcome those two barriers for these types of therapeutics? Maybe, I don't know, you want to start with Dave?
Let me start. You know, I'd say all very critical points, of course. Speaking for us, we're in phase 1, so we're still gathering information on just exactly what's going on in long-term responders and other responders who have a shorter response rate. So I'd say it's an interesting work in progress in our own portfolio, working within the contemporary T-cell engager world. You know, if you want to talk about improving durability as one example, you add a co-stim CD28. I think others have shown that. Regeneron's done a lot of work in here. I know J&J is looking at it with their Callican-2 program. But on our prostate cancer program, looking to further extend an already good durability profile, we're adding CD28. So I think where we're at is still early steps. I think similar to what others have found, we're going to find a variety of reasons: loss of target, mutations, immunosuppressive environments. We'll just have to figure those out as we go. For us right now, we're focused on the bispecifics. We'll see what that does with adding CD28, and then we'll go from there.
And how about you, Mathai? What do you think in terms of?
I mean, the data speaks. The Carvykti data, like people talk a lot about loss of target with BCMA. There was a whole effort around gamma secretase inhibition to prevent that. It turned out not to be necessary, I think in part because of things Roger's spoken about for a couple decades, that you don't need to get every cell. There is a form of in situ immunization that happens if you get a large portion of those cells in a tumor activating or killed by T cells. And so that's not been in practice a big problem. It does come down to choice of target. There are targets, of course, where the separation between tumor and normal tissue is insufficient, or certain targets that can, in fact, go away very rapidly. It turns out that certain ones like BCMA, CD19, 20, GPRC5D are not those targets.
Yeah, another question from the audience is, this relates to on that sort of the microenvironment that may be changing because of the target engagement you have is, how do you see potentially cancer vaccines in combination with these types of things? Are cancer vaccines could be something that you think are important for these combination therapies? Maybe Roger, you want to take that?
Well, first of all, I think it's important to emphasize, though I hate to do it in this forum, how little we actually know about the way in which immune activation ultimately results in tumors destruction. And so the simple observation is this: if you look across the totality of checkpoint inhibitor studies, what we know is that they can be quite effective, but the minority of patients respond. There are certain tumors in which the response is a majority. That includes melanoma, for example, a highly immunogenic tumor for reasons, again, we can't explain, but there are many hypotheses. MSI high tumors with a very high mutational burden, that may not be the responsible. I mean, the high mutational burden is certainly related, but whether that means you're forming new antigens is a whole different question. There, the majority respond, but in most cases, the majority do not. And the question is, what's wrong with those people who don't respond? And we ought to be able to answer that question, or at least I felt that way a dozen years ago. I still feel that way, it's just taking a long time to figure it out. Fundamentally, there are three things that could be wrong. There are probably more, but three make sense. The first is, maybe the tumor simply is unrecognizable by any immune system. There just isn't anything that the tumor produces that the immune system can recognize. The second possibility is, no, actually the tumor could be recognized by an immune system, just not by the patient's immune system. In other words, there's a hole in the repertoire of some kind, and they can't recognize the determinants on that tumor cell. And the third possibility is, well, actually, no, that tumor can be recognized and by the patient, but there's some other block, some other checkpoint that can't be overcome. Most of the work that's gone on has addressed that third possibility, trying to find things like LAG3 or TIM3 or TIGIT or other things that in one way or another would permit responses in those individuals, the majority, who don't respond. And I think we'd have to say that the yield has been poor. And one of the reasons why that's disturbing is because despite the fact that we've put a lot of years of effort, both in academic life and in industry, trying to understand exactly what the change was in the immune system as a result of checkpoint inhibition, we have not been able to my knowledge, identify a single patient in whom we could say, look, this was the immune repertoire before they got Keytruda, and then here's the repertoire afterwards, and here's how it changed, because we selected for these other high affinity clones, just as a hypothesis, and here's how they are, and these are the ones that killed the tumor, and we can show that in vitro. There's not a single example of that. We have no idea how that response is propagated. So it strikes me as a bit, premature to talk about heterogeneity of tumor cell clones, immune evasion, immunosuppressive microenvironment, all of those things for which we can generate evidence in preclinical models. But frankly, we still don't understand in a human system where you give Keytruda and you look at responders versus non-responders, what the difference is in terms of immune repertoire propagation. I think it's important that we nail that down.
Building on that, I remember AACR back in 2016 when they first presented the PSMA T-cell engager from Amgen. Mm-hmm. Your classic cold, add on top, cold immune environment, also called a desert. And I'm sitting in the audience going, this shouldn't be working. And so it just goes to highlight even beyond that, now graphed on top different cancer types. And yeah. I find it sort of interesting that that's one of the places, quite honestly, where the T-cell engagers are working. You could say it's because it's one of the first, but we've tried others. And is it because the tumor has done such a good job of evading the immune system over the years that it hasn't set up all these other barriers, so there's a lower threshold to activity with these T-cell engagers? So it's a really, really interesting point that you make. I think what it highlights here is, I view we're at the beginning of this.
Absolutely, no question.
And the learnings are exciting. Every 3-6 months, we're finding out something new in the literature.
Roger, I know what you thought about this some time ago. I'm wondering what your updated thinking is. There's a fourth way where the patient's immune system is capable of seeing that tumor or some antigen on that tumor. There's not a relevant checkpoint, but it's insufficient amount of that particular antigen. Hence, enter vaccines. Mm-hmm. And how important do you think that is? Sometimes, half the time, most of the time, that there's just insufficient levels quantitatively.
Yeah, I think that could be very important, and I don't see it as a fourth way. I see it as sort of a subset of the second. Mm-hmm. In other words, the patient's immune system either can see it but not very well because there's sort of a hole in the repertoire, and if you could just expand the repertoire, they'd be able to see it. Mm-hmm. And that could certainly be true. We do know examples where a cancer vaccine has been introduced in combination with a checkpoint inhibitor and produced interesting results. And that includes the work that's been done by Merck in combination with a vaccine created from a set of neoantigens that came from the Moderna team, and they're still working on phase 3 there, and similarly from the BioNTech group using a similar kind of approach. An immune attack on the tumor is a good thing, I think, probably under any circumstances. It may not be the case that the neoantigens that result from those individual substitutions are the important ones. And I say that because we do have good published data on a few T-cell receptor clones that have been isolated from patients with responses. And I think about the work of Mark Davis at Stanford, for example, where actually the thing that's being recognized is a monomorphic determinant that's present in other normal cells. And it's a subtle discrimination that's resulting in the recognition of the tumor population. So we should be careful to recognize that this goes beyond the interaction of alpha-beta T-cell receptor and its cognate ligand, and includes a whole set of other immunomodulatory processes that are going to need to be inventoried.
Okay. Just wanted to switch gears a little bit and talk a little bit about clinical trial design. There's been a lot of innovation in it recently, and I think one of the questions that come up is, what applications of technology can be used to enhance clinical trial design? As many of you are already aware, many oncology clinical trials are single-arm studies, so there's been applications of AI or other technologies to improve the scientific rigor. So, Mathai, would you like to take the question?
Sure. Like, this has been an objective for some time, that how do you judge a randomized control trial is the gold standard in many ways. Like, there are a lot of baseline features that you don't need to think too hard about if randomization is active. If you were to have a single arm rather than in addition a control arm, how do you judge that arm? And there's been a thesis for some years now that there's enough data in the domain of all interactions of patients with their physicians or the healthcare system. Can you draw enough out of there to judge that arm on its own? And enter the world of external control arms, or synthetic control arms, as they're sometimes called. And I'm of the view now that you can get pretty good, but you have to have a series of trials where you're developing this control arm properly. So, I had a very good experience with teclistamab, the BCMA CD3 that we mentioned before, where J&J at the time was running multiple myeloma trials, so Darzalex and Carvykti and others. And so the single arm, the dose expansion data came in from this particular compound, and to our eyes, who have been looking at a lot of myeloma data regularly, looked pretty good. And knowing those baseline features, we constructed a control arm. But what we did that was unusual is what you do when you construct such an arm is you select a bunch of features that you believe are going to be important, relevant to outcome, and you have to decide on how to weight them. There's a propensity score that you assign to each of those features, and we could compare our success doing that with comparison to a control arm in some other concurrent trial in multiple myeloma that we were studying. And so these adjustments were possible. We took that to FDA, and they were in fact convinced. We had put together a body of information that was strong enough that accelerated approval at a time in multiple myeloma where that was uncommon was in fact granted, and the drug was approved in 6 months. So it is possible to construct these, but to do so rigorously is hard. I still think it's worth, every time you run a randomized control study in any area, cancer, let's say right now, one should try to construct a third arm, so you start learning what features, what propensity scores are most appropriate, because all the subsequent indications, the subsequent medical affairs trials, the subsequent information that's collected on that will be greatly aided by an understanding of what would happen to a particular patient population. The majority of times it does not work well, but when it does, it's tremendously useful.
I think one of the issues, of course, is the quality of the data going in, and to be in the situation as you were at J&J, where you have a whole set of trials, and all of them done at GCP standard with a very similar set of metrics. it made this supervised propensity scoring, okay, not easy, but it could be done, right? The challenge is when you look out into the broader set of clinical data, it's a much less well curated set of data, and you can easily be led astray.
Yeah. And another big problem is that the measurements that are made in real life are quite different from the measurements you would choose to make in a clinical trial. I have advocated for using measurements that are in clinical trials that are closer to the measurements in real life, but there's a pretty significant gap. But when it's possible, one can try to do that.
Yeah, sometimes not possible because, again, of the distance between clinical practice and having linear scaling of something that you can actually measure. It's so difficult to identify things that you can measure that really matter, and that's a large challenge for the medical practice, of course, most importantly, but also for those of us who are trying to improve medical practice.
Yes. So we have another question from the audience. Compared to T-cell engagers, how do you think about in vivo CAR Ts? Is this really the future, or do you think T-cell engagers are still here to stay?
I think I commented on that already, but I think it has promise in the sense, it's worth trying in the sense that it is off the shelf, it is convenient, you can imagine access that's not that different from a T-cell engager, but there's a different set of issues to overcome. Can you transfect the cells that you wish to transfect? There's no purification step that there would be in an ex vivo CAR T, but there are a number of companies right now trying exactly that, so time will tell. We should ask that question again in about a year.
And then in terms of some of the things that you guys have talked about in terms of features that will help improve the sort of interpretation of trials, how do you sort of see where predictive biomarkers and novel predictive biomarkers play in that? Are they still important? Are they things that we don't need? Or are predictive biomarkers still important to try to develop from a research and development standpoint?
Well, we would all love to have them.
If they're predictive.
If they're truly predictive. But you can spend a long time wandering in the wilderness trying to find those biomarkers. I think that in the oncology space, there's reason to feel more confident that you can identify tumor eradication. And in most cases, the elimination of a population of cells that bears mutations that are associated with the transformed phenotype is an important indicator. So you can look for circulating tumor DNA, and that in many cases is a quite reliable predictor of the effect of therapy, and you can use that, as is done at many advanced centers, including here in Boston, of course, as a way of modulating, changing therapies in order to basically improve the extent of circulating tumor DNA reduction. And that gives you a measure, of course, of the tumor burden. It's not perfect, but it is measurable. It's a measurement that's largely linear, and you can infer results from it. But it's not yet the sort of thing, that's not easily reduced to practice except in very advanced academic centers. And we need a lot more early markers, both of tumor emergence under settings where we have a working therapy, but tumor clones, let's call it that, are emerging. We need more reliable things like that enable us to go in early and assess response to therapy. I think there's reason for optimism. There's just a lot of work to be done in the clinic.
I have grown more interested in circulating tumor DNA in the last few years because there's a few layers of information that are not yet fully tapped. There's the sequence information of ctDNA, and of course there's the methylation pattern. But one area that's not tapped much at all is that there's histone proteins that accompany the ctDNA, and those have marks on them. And there's all the normal marks that map closed and open sections of those histones.
Speak to expressivity.
Yes, and you can in fact come close to inferring a set of transcripts that are likely coming off that DNA. So it's more than the DNA sequence. You start to get into pseudo mRNA type or transcriptomic data if you were to mine that. And I've seen some good work recently there. So I do think that it's worth continuing to press on that blood biomarker because you don't need solid tissue. You don't need that biopsy. And as Roger said, you can monitor that little piece of very rich information for the duration of treatment and subsequently. So there's probably more to be done there as an important biomarker.
You know, we see it on our own programs, nothing elegant, but PSA and prostate cancer.
Sure.
Compared to our EGFR program, the rate of development of those two programs are night and day.
Right.
You
PSA
know,
is a pretty good marker
Yeah.
for tumor burden.
And so as a nascent biotech company, we had to pick our spots. The prostate cancer, there was proof of concept, early work from Amgen, Bayer, and others. We had a bloodborne biomarker. You know, we've moved beyond just depth, which people initially looked at, to how durable is that PSA reduction held. So we even see it internally. You know, our EGFR program, boy, I wish we had something to rely on that was anywhere 10x of what we get with PSA.
Right, and no one would embark upon a therapeutic program in prostate cancer without making those measurements. It's just absurd.
Yeah. And to me, that just reinforces, hey, where we started. We would all love to have them. Getting them is still the challenge.
Yeah.
Okay. We have a few minutes remaining, but I know we've been focused mostly on bispecific antibodies, but the panel did include combination therapies in the title, so make sure we want to touch on that too. So question we have is, what strategies have we seen and what lessons we've learned from past trials for combination work? You know, and what factors have determined where we actually see synergistic combinations versus overlapping toxicities?
And you're thinking specifically of checkpoint inhibitors, or
I think that's where
or very broadly.
Checkpoint inhibitors, but generally that's where most. Yeah.
I mean, I think that the checkpoint inhibitors, as we previously mentioned, there's been an awful lot of work looking for other checkpoints that are responsible for preventing an adequate anti-tumor response. And thus far, that's not been a terribly successful approach, although there are registered drugs. I mean, clearly, ipilimumab, Yervoy, is an active drug, and as well, there's some evidence of activity with LAG-3 active molecules, but it's not been the sort of thing that we'd hoped for that would reveal the pre-existing immunity in a much, much larger population. That is still elusive. I think the set of combinations, what we've learned from those, and this was tallied with great care by the late Emmett Schmidt at Merck, who died recently. What we've learned is that things that work in combination have single agent activity. It is very unusual. In fact, I don't think there's ever been an example of something which is not active by itself that is active in combination with a PD-1 directed checkpoint inhibitor. And the second thing that we've learned about them is that they work by an orthogonal axis. So it has to be mechanistically quite distinct to the extent that we understand mechanism. Those two things have permitted the identification of a number of promising new molecules which may be possible to use in combination with checkpoint inhibitors and give a much more dramatic result. In our own work at Eikon, we're deeply exploring those kinds of mechanisms, and with some success.
I mean, the question is broad, right? Like combination regimens across medicine within cancer, very broad. Maybe I can address it. You know, I feel in general, for most precision therapies, there's an under-investigation of future resistance mechanisms early on in the program, usually waiting for what might emerge, but to stress systems and push, see what might. And sometimes if you do that, you can start early with certain combination regimens that might give for more durable therapy. A good example of that is the drug that we developed a J&J called amivantamab. It's an EGFR c-Met bispecific antibody. c-Met is a resistance mechanism there, and cutting it off at the path, so to speak, seems to be effective, and it does give durability. There are other kinds of combinations that I think expose biology or do something more nuanced that actually can be remarkably interesting. My own company, there's a lead program that is a molecule that inhibits the interaction between beta-catenin and the family of T-cell factors, TCFs. And that's really interesting because there's a lot of important biology that happens downstream of that pathway that, if blocked, can give rise to particular combinations. So it, for example, would decrease stemness and correspondingly decrease the amount of DNA repair machinery, making more susceptible those particular cells to DNA damaging agents, and so on and so forth. There are a number of different things that can come from the dual efforts of understanding what resistance mechanisms might be, and the other is what biology sensitizes to what other biology. And doing so early in programs can be incredibly impactful to design.
I know we're out of time to say two things. You know, in our own internal, we're looking to add activity. We spoke about adding a CD28 bispecific on top of a T-cell engager to extend durability. You know, certainly there are those approaches that are important. Riffing on something that you just said, I spent a lot of time in antivirals, a decade or so, you know, where the turnover rate is just so much more rapid. What have we learned there? Coming to your point, if you've really got a targeted mutation, you know, where first, second, third gen mutations in some of these EGFR kinase inhibitors, if you applied the lessons we learned from HIV, HCV, where it's, you know, you didn't want to be on the trial with a singlet to doublet, you wanted to be there when there was a triplet. Those sorts of approaches, as they get rolled into oncology more and more, I think will be very important.
Yeah, one issue though to be aware of, and everyone recognizes, is, you know, the spectrum of parasitism. It's one thing to try and deal with a viral infection or certainly a bacterial infection where this all started, right? Yep. In combination chemotherapy. Cutting the difference between a normal cell and a tumor cell, for example, with respect to the sensitivity to things that influence stemness, that's a hard thing to do, because the parasitic population, I'll call it that, the tumor cell and the normal cell are awfully similar in terms of their behavior. Yeah. But that's for the future.
Okay. Well, I want to thank the time for the panelists, and importantly for the contributions you've made to the field of cancer. Thank you. Thank you. Thank you.